Hello, good afternoon, and welcome to the Porous Media Tea Time Talks. With these seminars, we want to give junior researchers working on porous media the chance to present their work to the scientific community around the world. So these talks are meant to be complementary to the geoscience and geoenergy webinars, which you can find in our featured channels. I'm Tom Bultrees from Ghent University. I'm here today with my co-organizers, Mohamed Noura from Oslo University, Catherine Spuren from Imperial College, and Maya Rucker from Imperial College. We're joined in spirit by Marcel Moura from Oslo University and Kamal Singh from Heriot-Watt University, who unfortunately cannot join us today. So with that, I'll hand over to Maya, who's going to chair the first session of today.
Thank you, Tom. Our first speaker today is AnnaMaria Eckel. She did her master studies in chemical and process engineering at Karlsruhe Institute of Technology in Germany and University College London, and started her PhD program end of 2018 at the Department of Chemical Engineering at Imperial College London. Her research focuses on convective dissolution on porous media with applications to geologic CO2 storage. The floor is yours.
Thanks a lot for the introduction. Also from my side, welcome to my talk. My presentation will cover some elements of my PhD project, which is about the study of solute mixing during density-driven convection on multidimensional porous media. Yeah, there is a growing concern over the global heating due to the increase of the greenhouse gas concentrations in the atmosphere, and it requires large-scale applications to reduce CO2 atmospheric emissions, such as carbon capture and storage. CCS involves the injection of CO2 into a porous rock formation, for instance, the saline aquifer, as shown here. At the interface between the injected CO2 phase and the resident brine phase, the CO2 dissolves into the brine, and the mixture has a higher density than the ones of CO2 or brine alone. And this leads to the initiation of convective currents, and the CO2-rich brine sinks in form of a finger pattern. One can expect that this density-driven convection enhances the CO2 dissolution rate into the water in comparison to a more slow diffusive mixing alone. And the effect can reduce the time in which pure CO2 could leak through fractures in the cap rock and increases the probability for a secure CO2 storage. It is important to characterize the effects this finger-induced stretching has on the mixing dynamics, and therefore we apply numerical simulations and are conducting experiments to predict the spread of CO2 and to support a successful sequestration operation. The strength of convective motion in comparison to diffusion is expressed in form of a Rayleigh number, which depends on fluid and reservoir properties. The appearance of the finger pattern differs between low and high Rayleigh numbers, as we can see in the concentration snapshots. In the following, I will first present some results from a computational study and will then proceed with the experimental work. In the numerical model, the saline aquifer is represented by a two-dimensional porous media, and we reduce the domain of interest and impose a constant concentration at the top boundary. Initially, the fluid is motionless and no solid is present. The governing equations are Darcy's law, the continuity equation, and the solid transport equation. Yeah, we make use of various mixing measures to characterize the mixing dynamics. So by regarding the contours of the plume, we can define the Isocontour lines at a 10% saturation concentration. To exemplify the spatial extent of the plumes, the spreading length is applied, which involves the displacement of the center of mass in vertical direction. And the overall state of mixing is described by the dilution index, which is associated with the increase in volume occupied by the solid, as well as the degree of concentration uniformity. We start with the results of the Isocontour lines. So on the right, we see the concentration distribution of Rayleigh number 10,000, and it is zoomed into a section right underneath the top boundary. The graph is showing the Isocontour lines versus the time, and in a purely diffusive case, the Isocontour stays constant over time. At first, the boundary layer remains flat, and the Isocontour coincides with that of a purely diffusive system. But after a while, a microscopic convective finger pattern is created, and as the fingers are forming and elongated, the Isocontour strongly increases with a growth rate of tau to the power of 1.5. After that, fingers are still formed and stretched, but simultaneously they merge with neighboring fingers, and the curve shows a reduced increase of the Isocontour. And eventually, the progressive saturation of the domain reduces the Isocontour towards 0. And the growth rate of 1.5 has been observed for all simulated Rayleigh numbers. The vertical spread of the plumes is characterized by the spreading length sigma and is plotted on the left. To express the temporal dilution state, we define the dilution index E on the right. At early times, the spreading length and the dilution index, they both grow to a power law scaling of 0.5, which is associated with a typical Fickian diffusion process. But as small fingers are forming and advection starts to be prominent, the scaling of sigma and E turns to a superdiffusive scaling of the power of 1.5. And it follows a regime of ballistic transport for the highest Rayleigh number in both graphs. Here it is important to exemplify that dilution, in contrast to spreading, does not include any information on the extent of the plume deformation. And despite this difference in the definitions, spreading length and dilution index both show a very similar behavior and a clear transition from diffusive to convective transport. To understand why we see these similarities in the vertical spreading and the dilution, we need to distinguish between effects provoked by advection or by diffusion. The former induces the stretching of the plumes, and it further determines the solute spreading. Molecular diffusion, on the other hand, dictates the diffusive mass transfer, and it primarily controls the dilution of the solute and therefore the dilution index. However, the diffusive mass transfer is intensified by the creation and the stretching of the plumes, which clearly affects the overall state of mixing due to an increase of areas of high concentration gradients. And therefore, changes in the spreading length and the dilution index are both characterized and dictated by the longitudinal finger-induced stretching. With this, I conclude the first part and will now provide some insights into the experimental progress. So available in literature are many two-dimensional studies, which are focusing on Healy-Shaw cells, which consist of two parallel plates, and in their gap, two fluids are filled to mimic the interface of CO2 and brine. The drawback of these studies is that they are completely disregarding the third dimension. 3D experiments on density-driven convection are still a novelty, but we are lucky because we have access to a CT scanner in the department, and therefore we are able to visualize the plume evolution in 3 dimensions. In order to do that, we fill a box with porous media saturated with water and place a thin foam on top, where paste made of salt and water is spread onto it. And due to a higher mixed density than the pure water, a finger pattern develops. And here we see an experiment with sand and on top the salt paste. Further, one can use glass beads and real rocks, such as sandstone, which we see here. And using real rocks or even sand will be very interesting, since no experiments with natural materials in 3D are available so far. In the shown CT image here, we used 100 micron glass beads, and the color bar represents the salt mass fraction. So at the top, the dark red represents the salt paste, and in the bulk, we only have water at time 0. Additionally, I extracted a horizontal layer right underneath the top, and there are no fingers at time 0. However, after 5 minutes, a pattern of small fingers is to observe, and after 20 minutes, the fingers have traveled downwards and are elongated, and even further after 40 minutes. And we can see between those two images here that merging between the fingers to a more complex pattern has taken place. And a video gives a more instructive view of the plumes, and by means of those CT scans, we will in future be able to investigate the temporal mixing behavior. Summarized in a few main points, we saw from the modeling work that density-driven convection does not only introduce convective mixing additionally to the diffusive mixing, but also strongly enhances the diffusive mixing concurrently. And further, we plan to perform experiments with homogeneous and heterogeneous porous media at various Rayleigh numbers, and we will compare the results with results from the literature. I would like to acknowledge a few people for their help and ideas, and this is my supervisor, Ronnie, then Rebecca, who is a postdoc at MIT, and Takeshi, who is a postdoc at Stanford. And my PhD project is funded by the Department of Chemical Engineering. Thank you very much for your attention, and of course, many thanks also for the chance to share some of my work today as part of the Porous Media Tea Time talk.
Thank you for that talk, AnnaMaria. It's very, very interesting. If anyone has any questions, please can they post them in the comment section of the YouTube video. I actually have a question. I was wondering, in your simulations, a lot of your fingers seem to go directly down. Have you done any simulations where maybe the porous medium is more heterogeneous and you have a more interesting shape to the fingering?
So you mean if we had this in the simulations?
Yeah.
So far it's only homogeneous, but we can always introduce heterogeneities into the model as well. So this is one step we might want to look at in the next step.
Are you waiting to have the experimental material before you do the modeling?
Probably we will first do the experimental part, because actually we're really interested in these 3D scans, because we think we have, that's one benefit we have to take this 3D. But then in comparison, we could always do also the numerical part to compare between numerical and experimental, but also between 3D and 2D.
Great. Are there any further questions from the committee or in the comment section? No, well, thank you again, AnnaMaria, for a really great talk. I now have the pleasure to introduce Thomas, who is a PhD student at Nottingham, Trent University in the UK. He uses 2D micromodels to investigate the effect of varying properties such as the spatial correlation, disorder, and deformation of porous media, and what impact this has on invasion patterns, which is what he will be talking to us about today. So without further ado, I shall hand over the floor to him now.
Yeah, thank you very much, Catherine, for the introduction, and thank you for the invite to present my this talk on my PhD. And as Catherine mentioned, it's looking at the effects of mainly disorder, but then how that is affected further by an elastic medium and spatial correlation, which I'll talk about in a minute. So just a brief outline of what the talk will involve. I'll give a quick introduction to the projects and some of the manufacturing we use. And the 3 projects have been the crux of my PhD, involve looking at this particle size correlations, how disorder has an effect on dispersive transport, and also then fluid flow in responsive porous media, and again, how disorder has an effect on that. So I'm sure this is a bit standard for everyone here, but just in case anyone's not sure, when we're talking about porous media, we're talking about a solid material that contains spaces or pores, and a fluid can enter into that, which obviously is could be something like a sponge, cement, or bones. And how a fluid flows in that porous media is determined by properties of the material and of the invading and defending fluids, so the viscosity of the fluids or the porosity of the material. And in particular, when I'm talking about disorder and particle size correlations, it's obviously important to remember that these pore throats here can vary the path that the invading fluid can take. So what has been shown before is that if you have a certain invasion, and this can be defined by the capillary number, so the viscosity of the fluids or the rate of invasion, if you keep that set and you say you keep the wettability of the porous media the same, then by introducing disorder, which can either be by random positioning of the particles or random size distribution of the particles, you can get completely different invasion patterns. So if you say look in this top left corner and you look in for different disorders, you get completely different patterns just by varying this disorder. And what we're investigating in these projects is how this has an effect by varying the properties of the system in different ways. So how do we make our experimental micromodels? We use a standard soft lithography technique. And what that involves is we have a silicon wafer and you spin coat it with some SU8 photoresist. And what that does is create a nice uniform layer. And then what you have is a photomask, which contains your designs, which has thousands of pillars on it. And these can be varied on a computer. You send that off and you get your photomask. And you shine UV light through that photomask and expose certain areas of the SU8 photoresist. And what this allows is to create a master mold, which is what you can see on the picture on the right. And what we do with that then is we pour PDMS on top of that, which is the kind of clear plasticky stuff that you might see on top of the master mold. And you can create thousands of these pillars. So this is an SEM image of one of our PDMS micromodels. And the great thing about lithography and this kind of method is what it allows you to do is you can choose where you want every pillar to be. You can choose what the radius of each pillar can be. And you can also, if you spin coat it at certain speed, you can also change the height of these pillars, which will be of importance in one of the projects I talk about. So the first project, as mentioned, was talking about something called the particle size correlation length. So what we mean by that is it's a length over which the particle sizes change significantly. So you can have a disordered system by having everything on a lattice position, and you have large and small pillars next to each other. So if you had a large pillar and a small and it was surrounded by small pillars, that would be a low correlation length because the particle size has changed significantly over a small distance. So as you have pillars of similar sizes around each other, you get a higher correlation length because they've got large pillars altogether and small pillars altogether. And we have, in this case, a radial system. And what we do is we fill this with a water glycerol mixture, and we withdraw the liquid through either the top or bottom outlet. And what this allows is the air to invade through the central inlet and propagate radially outwards, kind of similar to the figure A. So here's an example of one of the experiments for me to show you. So it's already filled with the water glycerol mixture, and what you see invading is this air in the white area. And what we do is we measure this until it eventually breaks out of the pillar area or the porous media area. And what we'll investigate is the amount of invaded area of the air. And also, if you can see here, there's some black remaining defending fluid. What we'd look at is how much of this is left over compared to the amount of total invaded area. So we did this alongside some simulations by some colleagues, Oshri Borgman, Ran Holtzman, and Enrico Segre. And here you can see a color map of what I was talking about. So you have low correlation length at the top row and high correlation length at the bottom row. So you've got a homogeneous system at the top, and you can see pink represents large pillars, and these are scattered everywhere. And then as you go to high correlation length, you get clouds of large pillars and clouds of small pillars. And our experiments worked in the low capillary range, so this 5 × 10⁻⁵ area. And the simulations were able to go to even higher capillary numbers than we were. And what you can see is that, especially for the higher correlation length, the flow seems to invade via the small pillar areas as you'd expect. And our experiments seem to follow a similar pattern. So what was interesting then is when we looked at the saturation or the amount of invaded area and the trapped fraction, so how much defending fluid was left behind compared to the invaded area. And the correlation length seems to have a significant effect at the low capillary regime, as you can see here. So at low correlation length, you get a lot more saturation. As you increase that correlation length, the amount of saturation decreases. But there's also a balance in that at the low correlation length, okay, you invade more area, but you also leave more trapped fluid behind. And then this is reduced again for higher correlation length. But what was actually surprising with this project is that even in the high capillary regime, where viscous fingers dominate the kind of invasion pattern you see, correlation length still has an effect at this point, even though it's less significant. And if you want to know more about this project, there is a paper currently published on this. The second project, again looking at disorder, involved dispersive transport in porous media. And this followed on from some work by our colleague Karen Alim, where if you have a system of pillars, and then you can imagine the space between those pillars as tubes, what you have is then a network of tubes. And as you increase the disorder of the system by randomly varying the position of each pillar, you then change the tube sizes and the tube angles. And what she showed was that it's the standard distribution of these tube sizes that actually affects the overall flow distribution. And we followed on from this by doing experiments looking at dispersive transport in these kind of systems. So what we do is we have our PDMS system again, and this time it's rectangular rather than radial. And we fill it with some plain water, and we inject some fluorescein dyed water with a known concentration, as you can see here. Just like this. So this is a time-lapse image. And what we do is then we take intensity line profiles along the length of the chip. We take more than this amount of lines, it's usually about hundreds. And if you've calibrated the system, you can know from the intensity of certain areas what the local concentration is at that point. And this is now still ongoing. We are finalizing the experimental results for this. But what seems to be happening from the numeric side, at least, is that there's a non-monotonic trend in the width of that front. So if you measure along the front of the fluorescein dyed water invasion, you get the variance of that first decreases with increasing disorder, but then actually increases again with increasing disorder. So what seems to be happening is that as you're increasing the disorder, you're starting to get mixing between the advective lanes. And then as you go into higher and higher disorders, you start to get diffusive islands where the fluorescein is only invading via diffusion rather than advective flow. And we're just verifying this kind of trend now with experiments. So there's still some bits to go on this. Finally, the last project that I'm currently working on, again, we are finalizing results for this, was looking at fluid flow in a responsive porous media. So what normally happens in a rigid medium is you might get some kind of invasion pattern looking like viscous fingering or capillary fingering. But what can happen is when you allow your particles or pillars, in my case, to either deform or be displaced, you get a new pattern emerge called fracturing, which can be characterized by these thin, long fingers that propagate outwards quite quickly. And we investigate this by having another rectangular system of pillars, and we allow these pillars to bend. And then we look at, okay, when we see this fracturing pattern, how is this restricted due to the effect of adding disorder into it? Now, we had to be a bit creative with how we did these systems, because using our soft lithography method, there's only so tall we can make the pillars, and therefore so much it can bend under the invasion pressures. So what we instead did is we created a PDMS well, such as this. This is a bit exaggerated, but you create a well that's quite shallow, and then you have your system with your pillars, and you place that on top of the well, such that the distance between the bottom of the pillars and the bottom of the well is small, and invasion is still preferred via in between the pillars. And what this means is that you can get much more elastic pillars than if you had them fixed at both ends. So some of the patterns we see are so on the right hand side, you can see a low disorder system where the disorder is only introduced via manufacturing error, which is about 3 or 4%. And you again see these kind of long, thin fingers, which propagate quite quickly. And then if you introduce disorder by randomly varying the size of each pillar, you then get this fracturing pattern seems to be restricted, and you get more something like capillary or viscous fingering at this point. So this is exactly the same injection speeds, exactly the same height pillars, but a different rate, a different disorder. Now, I'd say this is still ongoing, but what, as a little teaser for in the future, hopefully, is that what's interesting is that we seem to find that these kind of fracturing patterns only occur during a constant flow rate experiment rather than a constant pressure experiment. So what we see again is where you have these hanging pillars and you do a constant flow rate, you see again these long thin fingers, but then as you do a constant pressure by having a reservoir a certain distance below the chip and keep it at constant pressure, this fracturing pattern seems to disappear. And this is what we're investigating at the minute. So just to conclude, 3 projects. We investigated the effect of disorder in 3 systems. And this can be by correlation length, looking at dispersive transport, and also when the porous media is elastic. What we find for the correlation length is that at high capillary numbers, the effect of the correlation length is minimalized, but still in effect. But then at low capillary numbers, it's even more significant, and there's a balance between the amount of saturation and the amount of track fraction left behind. For dispersive transport, when you introduce disorder, you find a non-monotonic trend, such that the width of the invading front is reduced at first when you increase disorder, and then as you get to higher and higher disorders, that width is increased. And this is due to the emergence of lane mixing and then diffusive islands, like I said. And then finally, we've looked at an elastic system, and you find that if you allow the pillars to bend, you create a fracturing pattern, but this is restricted for when you add disorder into the system. And what we're also now looking at is, as I say, constant flow rate versus constant pressure. There seems to be a difference, and we're investigating this. So just some acknowledgements quickly. My supervisory team, Lucas Goering, is my director of studies. My co-supervisor is David Fairhurst, and my independent assessor is Carl Brown. Some of the people who've worked on the 3 projects with me, Oshri Borgman, Rand Holtzman, and Enrico Segre. Karen Lim, as I mentioned, Felix Meigel and Leonie are currently working on the dispersive stuff. And then a quick shout out to my lab team, who have given me a lot of support throughout the years. Thank you all for your attention. If you have any questions, please ask, and my email is at the bottom here.
Thank you very much for the great presentation. The talk is now open for questions. If you have any, feel free to post it in the comment section. We will then read it out and pass it on. For now, I actually do have a question. I wondered a bit, so you used the pillar size to calculate the correlation length. Is this correct? Have you considered also to look into the pore sizes rather than the pillar sizes? Also because when you have, of course, change in your structure due to load, it might change the pore sizes.
Yeah, I agree. So what we basically did, just to give an outline of how we generated the stuff and how we came to using particle correlation, is basically what was done by the numeric team who decided on using who decided how to calculate the correlation length. They basically made a Gaussian distribution of possible pillar sizes, and the width of that distribution set the correlation length. So if you've got a large distribution, it means the pillar size could change quite significantly from one pillar to the next, whereas a smaller distribution. So we basically just used that to generate the pillar sizes, and that's how we then looked at the correlation length. So the pore sizes themselves, we didn't look too much into. It was more just a case of we varied the possible pillar sizes from one to the next, and then look at that effect.
Okay, well, you wouldn't expect the change in the shape of the pillars to be somewhat causing the effects which you see in terms of the patterns.
Sorry, what do you mean by the shape of
the flow patterns? Well, if you put on load on them, then of course they change the shape
so
in a way.
Basically become an ellipse slightly due to the pressures. We didn't look at that so much. I don't think we considered that. I can't say from experiments. So we normally, when we did the experiments, used a microscope to see exactly what's happening with the pillars. So we could look at different sizes, and we make them so small, if you like, the aspect ratio so low, such that the pillars can basically be considered rigid. I mean, there will be some effect, some slight effect of the evasion causing some slight contraction of the pillars, but I think it should be so small an effect not as to be significant. If that answers.
Okay, thank you. This answers my question. I don't see any questions in the comment section. So if there are any further in the committee.
I'll ask a question. Sure. So in the slide you showed about the constant pressure versus constant rate. Yeah, so is this something, I mean, the pressure that you worked at, I think it was 5,000 pascals. Was that enough to bend the pillars?
So I haven't shown it. Just for reference, I'll skip to it so people can see. This is actually at constant pressure, this video here. This is saved a bit. And it might not be obvious that the pillars are bending here. I'm not sure how well you can see this. But if you do a difference image between each frame, what you'll see is that as the fluid invades, you can see white areas around the pillars. Can you see that? So because it's a difference image, so you've got your pillars before invasion, your pillars as it's being invaded, you can see the pillars being deformed due to those white flashes. So we do actually see that that pressure is enough to deform the pillars. I don't know if that's obvious from the video.
Yeah, you can see it kind of, I guess. But the other thing I was wondering about is that one of the differences in drainage is that in constant pressure, you wouldn't expect Haynes jumps, though you can kind of see Haynes jumps here, I think. Yeah, so which might be due to, I don't know, size effects or whatever. But I was wondering if that could be another reason for why you see a difference.
Possibly. What I will say is that this is like, this constant pressure is not something we expected and is actually quite new and is what we're investigating at the minute. But even with the experiments not showing it, we also have done some simulations, and even in the simulations, you don't see any fracturing patterns due to elasticity. It doesn't occur in the simulations for constant pressure. It's only really when you introduce constant flow rate that you then see this effect. And what the reason for that is we're not sure yet, but this is what we're investigating.
Okay, thank you.
Okay, so if there are no further questions, I would like to thank both speakers again for the great presentations today. Our next session is 3rd of November with Ankita Singh and Musa Hossain Mir. So I hope to see you again in 2 weeks' time. Thank you very much for listening today. If you have any questions or comments or anything, please just send us an email or post a comment in YouTube. Thank you.